Changes such as mutations in chemicals and signal transduction pathways can alter cellular responses. So, as we look at this, we have our receptor and we have the signal transduction pathway within the cell. So, you have your signal, and it's going to land on the receptor, and it's going to cause a change or a cascade of events, and we're going to get responses. This pathway leads to this response. This pathway leads to this response. This pathway leads to that response, and that one leads to that response. So again, we're getting multiplicity here. So one particular receptor can cause multiple responses. So now if we look at some changes or chemicals or mutations, we're going to see how that affects the whole sequence. So in this case, we've got this inhibitor, and it's going to land on the receptor site, and then the hormone or the signal or the ligand can't bind, and that's going to cause everything to not work, and we get no responses. In this case, maybe the receptor itself had a mutation. If that mutation is existing on the site of the receptor where the ligand is supposed to bind, again, nothing happens. Now, if the mutation is on the intracellular domain of this receptor protein, you're still going to have the ligand be able to bind. However, because this is mutated, then nothing else would work interiorly, and you'd have no responses, even though you have hormone release and you have reception. So, as we keep looking here, we're going to think about this idea of downstream. So you may be told where the problem is. In this case, it's this E4, this enzyme that's helping convert A into E. So what works and what doesn't work? This is going to land, the reception works. That can still transfer, and all of these responses are going to still happen because the problem is only right here, which is going to cause the first response to not occur, but all the other responses would still work. Another example here, if we messed up this particular part of the pathway, what still works? That works, that works. We get response number 1. This still works, and we get response 3 and 4. However, we do not get response number 2. So again, understanding and pinpointing when you look at a signal transduction pathway, what is working and what is not working is the goal here. So sometimes we don't just have mutations in particular proteins or DNA that leads to the malfunctioning of certain proteins, but we can have chemicals. And a lot of times you see this in these diagrams, and this symbol right here means it's interfering and stopping this from working. So we have this working, that working, and these working. However, this chemical is interfering with this particular step, so we do not get response number 4. As we keep going here, other ideas. Most things, their natural state, if the signal is not being sent, we don't want to do any of these responses, right? So it's only some kind of stimuli in the environment signals the organism to actually start a pathway. So we're not doing the pathway all of the time unless the message is being sent and there's communication that the pathway is supposed to happen. So typically this would be the original setting. If there's no signal, there's no response. The transduction pathway does not occur. However, there can be mistakes that occur where even though the signal isn't being sent, a mutation in this process can cause the responses to still occur, even though they don't have the message to actually do it, which is a major problem, because if you're only supposed to be responding in the presence of some stimulus that causes the organism to release a signal, you don't want all of these things occurring. unless you need them to occur. So another idea, especially when it comes to cancer, sometimes we have, imagine this is a cell and this is empty space above it. Now, if there's empty space and cells are supposed to be touching, and there's a cell that dies and there's a gap, you can have this receptor creating the responses to cause this cell to grow or divide and fill in the empty space. Now, in this kind of situation, it's supposed to grow and have all those responses unless another cell comes in contact, and then it should not grow, it should not divide, and now this, because there's contact here, you should not have any of these pathways. In the case of cancer, sometimes you have that cell touching and it should be told don't grow, don't divide, but there's a malfunctioning in those cancer cells where even though this pathway says don't do it, it still does it and it still divides, even though because it's not responding to that contact from the other cell. So looking at some other examples, we've talked about antidiuretic hormone before. Again, we have this pathway, antidiuretic hormone comes in, you have the signal transduction pathway, and ultimately you reabsorb water from the kidney tubule back into the blood. So alcohol, though, inhibits antidiuretic hormone. So alcohol is considered a diuretic. If it inhibits this pathway, can you kind of imagine what's going to happen next? Ultimately, antidiuretic hormone would not be able to land and we would not reabsorb that water. So ultimately, what's going to happen as you have alcohol in your system, you're going to pee out a lot of dilute urine. Okay, another example here, we're looking at glucagon. We understand with glucagon, the signal transduction pathway and phosphorylation cascade is meant to break down glycogen and ultimately get that stored energy reserve of glucose back into the blood. So if we look at this, again, you see this symbol, which means it kind of is interfering or stopping. So this proposed model says that metformin, right here, enters the cell and acts on the mitochondria, causing an increase in AMP. Elevated cellular AMP levels inhibit membrane-bound adenylyl cyclase, causing a reduction in cellular cyclic AMP or CAMP levels and decreased pKa activation and target phosphorylation. Again, we have all these big scary words, but if you can read a diagram and then visualize what's happening, you can kind of understand what's working and what's not working. So again, if metformin comes in and acts on the mitochondria, which increases AMP levels, and AMP is going to interfere with adenylyl cyclase, the membrane-bound enzyme, what still works? So you're still going to have reception. You're going to still have this G protein transfer, this G protein coupled receptor, and you have your GTP landing, and this is moving. However, because this is interfering with adenylyl cyclase, you do not get cyclic AMP, and now everything downstream of cyclic AMP is not going to work, and you're not going to raise your blood sugar when you have metformin in your system. So from this new understanding of metformin's action, Birnbaum and colleagues surmised that adenylyl cyclase could be a new drug target by mimicking the way in which it is inhibited by metformin. This strategy would bypass metformin's effect on cells' mitochondria to make energy and possibly avoid the adverse side effects experienced by many people who take metformin, perhaps even working for those patients resistant to metformin. So now we have a new drug instead of metformin, and metformin acted on the mitochondria. This new drug, though, again, is trying to bypass the effect on the mitochondria and directly target adenylyl cyclase. So again, this enzyme is taking ATP and turning it into cyclic AMP. So again, it's trying to get involved here in that same way. So what works, you still have this. However, again, you're not messing with the mitochondria this time. We don't have increased AMP levels, and you just have the inhibition of this particular enzyme. So with all of these examples, you have changes, a lot of times through mutations or chemicals, that can alter cellular responses through signal transduction pathways.